Thank you to the organizers for asking me to speak, and I'm going to talk about two somewhat related topics today: clinical trials and also immunotherapy for sarcomas. And essentially, what are clinical trials? And they're basically experiments to assess the value of a treatment. And basically, all anti-cancer drugs currently available on the market have gone through the clinical trials process. So all of the drugs that Dr. Rattan spoke to you about were once investigational drugs before they got licensed for use. And this process is designed to select drugs which are both safe and effective, and also importantly, to protect the public from ineffective or potentially harmful agents. And these are therapeutic drug trials. There are also clinical trials that can test for the impact of, for instance, lifestyle changes on certain disease states, or to look for markers of disease in blood or tissue specimens. And those are non-therapeutic trials, and we're not really going to be speaking about those today. Okay, why should we bother with clinical trials? Well, the alternative to clinical trials is the anecdote. And we've been through a lot of that in the last year and a half with, I heard drug X works great for the coronavirus, without any real formal assessment of the drug. And clinical trials for new drug approval are an arduous process, and only 3.3% of drugs that enter the clinical trials process eventually become approved agents that come to the market. And that number improves when the drug has a specific target, as Dr. Ogolnik was alluding to. But even in that situation, the success rate is still around 10%. So at least 9 out of 10 drugs that go through this process don't make it through successfully. And the costs for this are quite substantial. And the cost of bringing a new drug through the approval process is estimated at close to $650 million, with a range up to close to $2 billion. And it's a time-consuming process, and the median time to develop a new cancer drug is over 7 years. And what are the phases of clinical trials? Well, the initial study of a drug is what's called a phase 1 trial. And those are the initial trials for drugs in people. And the agents are selected based on preclinical evaluation in the laboratory. And often these are first-in-human studies, which means that human beings are getting these drugs for the very first time. And they typically start with a low dose based on information that's been gleaned from the animal studies. And a phase 1 study will include dose escalation to look for any unacceptable side effects and to find a recommended dose and schedule for a phase 2 study. And it's called the RP2D, the recommended phase 2 dose. And phase 1 studies also look to see how an agent is metabolized and distributed in the body. And an important thing to realize is that phase 1 studies are not designed to test for efficacy for the drug. However, approximately 6% of patients on phase 1 studies actually do derive a clinical benefit. Okay, and phase 1 studies are generally small trials with a limited number of participants. And in the past, these trials were open to a wide variety of patients whose disease, whatever disease it was, had progressed on all known active agents, who had a disease that had no established effective treatment. And therefore, a phase 1 study might include a whole grab bag of patients with carcinomas like breast cancer and lung cancer, lymphomas, sarcomas, and melanomas. And with the advent of targeted therapies, some phase 1 trials now limit participation to subjects with a known or suspected target. And that potentially now limits access for sarcoma patients to some of the newer clinical trials. But as Dr. Ogolnik also alluded to, some trials are now disease agnostic, and they include all disease types with a specific marker. And a recent example of that is, patients whose tumors have what are called NTRK fusions were entered in trials irrespective of what their primary tumor was. And there were carcinoma patients mixed with sarcoma patients, and they all, if they had that fusion, that were likely to respond, and several drugs in that class have now been approved. Phase 2 trials are trials which build on the knowledge gained from the phase 1 trial, and they test the agent generally against a specific disease or target. And these trials seek to determine the efficacy of the drug and generally include a larger number of patients than phase 1 studies. And there's sometimes studies that are sort of combined, and they're phase 1b/2 studies. They're designed to go seamlessly from phase 1 directly into phase 2. And these studies are used to make what's called the go-no-go determination of whether you should proceed with a phase 3 study. And this is where a lot of drugs get hung up, and where some drugs make it through phase 1 and phase 2, but unfortunately don't succeed in phase 3. And phase 3 are larger trials designed to test the new agent against something else. It's either the existing standard of care, if one exists, or against placebo if no standard therapy exists. And they're designed to determine if the new agent is better than what we had before. And the better will be designated in the trial. It will either be progression-free survival, overall survival, a lessening of toxicity, or an improvement in quality of life. And phase 3 trials are generally larger. They're expensive trials, and they can often be multi-center or multinational. And successful completion of a phase 3 trial is used to obtain regulatory approval for the agent to be marketed for use. And the way we assess response in oncology with solid tumors is something called RECIST, and it stands for Response Evaluation Criteria in Solid Tumors. And a complete response is basically the disappearance of all tumor. A partial response is at least a 30% reduction in the diameters of the target lesions of the tumor compared to baseline. And again, no new areas. And progressive disease is at least a 20% increase in the sum of the diameters of the target lesions, or new lesions being seen. And stable disease is when you're in the middle, and it's not enough for a partial response and it's not enough for stable disease. And in recent years, there's been recognition of something called the clinical benefit rate, which combines complete responses, partial responses, and stable diseases. And particularly in patients with GIST, that's been found to be a very useful ratio. Because patients with stable disease often do as well as those who've had a complete response. And this is just an example of a phase 1b/2 study that was done looking at Doxorubicin with a drug called Olaratumab, which we thought was a targeted agent. And in the upper panel, you can see the red curve is the combination of Doxorubicin and Olaratumab together. And it's looking at progression-free survival. And the progression-free survival with Doxorubicin is in the blue line, and the red line is superior to the blue line. And this study had about 130 patients in it. And the progression-free survival was modestly improved. But when you look at the lower panel, which looked at overall survival, there was a really dramatic improvement in overall survival of close to a year. So it went from 14.7 months to 26.5 months, just short of an entire year. And this was so dramatic that this was submitted to the FDA. And on the basis of this phase 1b/2 study, the FDA approved the drug conditionally and said, we'll let you put the drug on the market, but you have to do a confirmatory phase 3 study. And unfortunately, these are the results from the phase 3 study that showed, in terms of overall survival, the two curves are really superimposable, and the Olaratumab did not really show any added benefit to Doxorubicin alone. And they looked at subgroups in this study, also looking at leiomyosarcoma groups, and they saw no difference there as well. So the conclusion of the phase 3 study was different from the phase 1b/2 study, and it showed that adriamycin was still the gold standard. Olaratumab has subsequently been withdrawn from use, except for people who were on the studies and deriving a benefit from it, or looking at it in other situations. So this was a disappointment, but it sort of proved the value of going through the process. Okay, phase 4 trials are conducted on already approved agents and may look at issues such as long-term effects or other factors like the color of the pill. What are the components of a good trial? Well, you have to begin with sound science and a good idea for what you're trying to accomplish. And good trials are generally free of biases, and biases can be unconscious biases, but the things that are usually hallmarks of good studies are randomization, so that patients have an equal chance of getting into either arm, whether it's a 1-arm or a 2-arm or 3-arm study. It's also helpful when you see that studies are either blinded, which means that the patient doesn't know that they're getting which drug they're getting, or double blinded, where neither the patient nor the treating physician knows which agent they're getting. We talked a little bit, and others have talked about placebo control. That's useful when there's no effective known treatment. And it's sometimes a barrier to entry into studies, but allowing for crossover at the time of progression can sometimes improve that. Another hallmark of good studies is there can be independent central review. So the doctors who are conducting the study are not the ones who decide whether they're seeing responses or not. Okay, clinical trial participation. Unfortunately, only somewhere between 2% and 7% of adult cancer patients enter clinical trials in the United States. And this is in contrast to childhood cancers, where approximately 60% of children enter clinical trials that are usually sponsored through the Children's Oncology Group. And it's not clear why there's such a big difference. Our pediatric oncology colleagues attribute it to the fact that they're better doctors than we are, and that may be part of it. What are the advantages to clinical trial participation? We can get access to new treatments before they're commercially available. And generally, the clinical trials will be run by doctors who are very knowledgeable in the specific disease, and you'll have access to experts. And the other thing that's important is altruism. It's the goal of helping others in the future to move medicine forward. And this is particularly true for phase 1 studies, where efficacy is not usually the endpoint. The disadvantages of being in a clinical trial: the new agent can have side effects or unacceptable toxicity. The new agent may be inferior to existing agents, or it can even be inferior to placebo. And we've had patients on placebo-controlled trials, and the patients on placebo do better. And you may wind up on the placebo arm in a study with no crossover, so you're not actually getting any medication. Barriers to clinical trial participation. There can be structural barriers. For instance, there's just no trial available for your stage of disease. There can be clinical barriers where there is a trial that you're eligible for, but your kidney function, your liver function, your heart function is below the required entrance criteria for the study. There can be physician barriers where your doctor either isn't aware of a particular study, or for whatever reason doesn't feel that you would be a good candidate for that study. And that's where sometimes bias can come into the way studies get performed. And there can also be patient barriers to entry into clinical trials. Some of that can be in desire to determine your own treatment, feelings of loss of control, patients not wanting to be guinea pigs, fear of unknown side effects, concerns about costs. There was a recent article in one of our journals about the financial costs of going on to clinical trials. And finally, there are logistical barriers. You know, you may be living in Idaho and the trial is in San Francisco, and you don't have the transportation to get there, or there are sometimes ways to get around that. And particularly for trials at the NCI, they will often defray costs for you to go on studies there. Okay, and how do you find a clinical trial? Well, this is the clinicaltrials.gov website that was alluded to previously, and you can look at the rectangles in the middle where it says condition or disease. You can just type in your sarcoma. You can put in potentially what stage it is, and you can select which country, and for us it's the US, and it will give you a list of potential clinical trials. And there are different filters once the different pages come up, and you can hone down to filter to see just which trials are actually open and accruing patients. So this is something that you have that either you can do on your own or you can do with your physician. All right, so we're just going to switch gears now and talk a little bit about immunotherapy. Immunotherapy is not a new concept. It's the goal of harnessing the body's immune system to fight against malignant tumors. And the picture on the right is Dr. William Coley, who was the head of the bone tumor service at Memorial Hospital in New York. And he joined the staff there in the 1890s, and he heard of a patient who had a malignant head and neck tumor that got better after the patient developed erysipelas, which is a localized skin infection. And he examined the patient, and he found the patient had no evidence of cancer. So he felt that somehow the body's response to the skin infection had helped fight against the tumor as well. And he began actually initially injecting patients with live streptococcus, and this proved unfavorable. And he then altered the process and gave a combination of heat-killed streptococcus and Serratia marcescens, and this became known as Coley's toxin. And this was given to many patients over the years, gradually fell out of disfavor, but it sort of proved it sort of supported the idea that you could use the body's own immune system against tumors. And there are three potential ways to target the immune system. You can use what are called immune checkpoint agents that are designed to essentially untether and unleash the immune system. And the common ones that are in use now are PD-1 and PD-L1 agents, or antibodies against CTLA-4. People are also looking at trying to make vaccines that will target something specific to the tumor cell. For instance, there's a marker NY-ESO1 that's seen in synovial sarcomas and some myxoid liposarcomas. And the last way to use the immune system is to essentially create cells or alter cells that will be better at fighting tumors. And there are several of these listed. There are CAR T cells. The CAR stands for chimeric antigen receptors. There are T cells that get transduced with T cell receptors. And the latest are what are called SPEAR T cells, which are specific peptide-enhanced antibody receptor T cells. And this is just a little cartoon from the pembrolizumab folks that shows the T cell in the lower left portion and the antigen presenting cell, and how the PDL1 normally binds to, or PD-1 binds to the PD-1 receptor and inactivates the immune system. And by giving an agent such as Keytruda, you block that process and you essentially unleash the immune system. And this, I apologize, is a very busy slide, but this looks at recent results from immunotherapy trials using these three different modalities. And you can see in the upper third, the checkpoint inhibitors, the agents or drugs, ipilimumab and pembrolizumab and nivolumab are either PDL1 or CTLA-4 antibody constructs, and they have some activity in sarcomas, although the activity is not dramatic. There are some tumor types that really have no response. The largest response was seen in the Taube trial in undifferentiated pleomorphic sarcomas with a 40% response rate. But you can see that the median progression-free survival was modest at 18 weeks with an overall survival of a little less than a year. If you drop down a little bit to the Wilkie trial, that trial combines axitinib, which is a tyrosine kinase inhibitor, with an immune checkpoint drug, and they saw a higher response rate in all their patients, which was 25%, but they in particular saw a response rate of over 50% in patients with a rare tumor called alveolar soft part sarcoma. And we'll talk about that a little bit later. The adoptive cell therapy is generating T cells that will fight against your tumor. And there have been several studies showing activity. This is one of the earliest ones from Dr. Rosenberg's group, showing 4 out of 6 responses with T cells that were altered to target NY-ESO-1 on synovial sarcoma cells, and they saw 4 out of 6 responses. The vaccines have generally been disappointing, except for one trial that used radiotherapy with intratumoral injection of dendritic cells. And we would need to see more studies along that line. This is just going into a little more detail about one of the largest studies that was done by a cooperative group that many of us belong to called SARC. And it looked at pembrolizumab, which is a PD-L1 agent, and they looked at advanced soft tissue and bone sarcomas, and it was an open label phase 2 trial. And this is the breakdown of the patients. They saw it was 40 soft tissue tumors, 40 bone tumors. And they looked at specific groups. And unfortunately, they did not include a group of just other, a grab bag group. So it was lyomyosarcomas, sarcomas, UPS, liposarcoma, and synovial sarcoma for the soft tissue tumors. And for the bone sarcomas, it was chondrosarcoma, Ewing's, and osteosarcoma. And they saw one complete response with an undifferentiated pleomorphic sarcoma, and they saw a smattering of partial responses, but the overall response rates were low, although there were a significant number of patients who had stable disease. And this is just a waterfall plot that shows the responses with the bone tumors. And you can see the rectangles that go up are progression, the rectangles that go down are decreases. And there were 2 to 3 patients that met the criteria for partial response. Also bear in mind, if you look on the left at the leftmost column, the pink column that goes up above 500, there were several patients whose tumors grew significantly on these drugs, and we'll talk about that a little bit later. And this is just the waterfall plot showing the responses with the patients with soft tissue sarcomas. And again, you can see that the sort of kind of red-colored rectangular bars, that the best responses were seen in undifferentiated pleomorphic sarcoma, although the patient with the greatest increase in size, again on the extreme left, was also a patient with undifferentiated sarcoma. Okay, so immune checkpoint drugs do appear to have activity in some tumors, but not as dramatic as what's seen in melanomas or in some patients with lung cancer. But there are some rare tumors that seem to have particular sensitivity to these drugs, and they unfortunately were not included in the SARC-28 trial. And these include cutaneous angiosarcoma, chordoma, and alveolar soft part sarcoma. And it's not exactly clear why these sarcomas tend to respond better, but this is just an example of one of our patients who was given pembrolizumab for a progressing cutaneous angiosarcoma. You can see on the left was his tumor around the ear in July of 2019, and he had been through essentially every active agent for angiosarcoma, and this tumor would periodically bleed, causing him to pass out. And he would be found by the paramedics in his kitchen with blood splattered all over the place, like in a slasher movie. And, within several months of starting on pembrolizumab, the area just gradually improved and stopped bleeding. And this is from March of 2020 on the right-hand panel. You can see that there's essentially complete resolution of the area. which continues to this time. The toxicity of immune checkpoint agents, they're actually usually quite well tolerated compared to chemotherapy, but there are side effects because you're essentially unleashing the immune system to go after these tumors. So you can see pneumonitis, which can manifest as either cough or shortness of breath. You can see colitis with diarrhea or abdominal pain. You can see nephritis, it causes abnormalities in kidney function. You can see immune endocrine disorders with problems with the thyroid or the pituitary or the adrenal gland. You can see cerebritis, which is an irritation of the lining around the brain, or actually in the brain, and you can see confusion sometimes and memory problems. And you also need to exhibit care when you give these drugs to patients with pre-existing connective tissue diseases like lupus or rheumatoid arthritis, because you can sometimes actually make those symptoms much, much worse. And some of those patients are just not able to tolerate these agents. There are two other things to bear in mind about immune drugs. There are two concepts. One is pseudoprogression. And pseudoprogression is the temporary enlargement of a tumor following administration of an immune checkpoint drug or any immune agent. And it may be related to an influx of immune cells into the tumor region. They're actually fighting against the tumor. And it's important to recognize that this may be happening because this will generally subside with time. And this may be actually a sign that the patients are going to respond to treatment. And this was discovered in the early trials with patients with melanoma, where they were taken off study because it felt that they had progressive disease. And they came back for follow-up several months later, and it was observed that their tumors were much smaller. So people began to realize that pseudoprogression was a real phenomenon. There's another entity to be aware of, which is hyperprogression, which is the paradoxical accelerated growth of a tumor after exposure to an immune checkpoint drug. And this may occur in up to 30% of cases. It can be associated with patients with older age, tumors that have MDM2 expression, which happens in sarcoma, for instance, in liposarcoma and other tumors like pulmonary intimal sarcomas. And it also appears to be related to prior exposure to radiation. And the mechanism for it is not well understood, but it's something to be that we need to be aware of. So the future of immunotherapy for sarcomas. Well, first of all, we need to have a better understanding of the science. We need to better understand which tumors may be able to respond to these agents. And as Dr. Ogolnik, alluded to, patients whose tumors have high mutational burdens are now approved to get these drugs, regardless of their tumor type. But there are some tumors that are considered hot tumors with many mutations, and other sarcomas are considered cold. And one of the areas of interest is there a way to turn a cold tumor into a hot tumor so it can potentially respond to these drugs? And the carcinomas, which tend to respond well to these agents, often have high mutational burdens. And many sarcomas, as I said, have low mutational burdens. And rather than having mutations like the carcinomas, they have amplifications of genetic material in their cells, or they have translocations where material is switched, but it doesn't appear as a true mutation to the immune system. So in the future, I think, and people are doing this now, as shown in the Wilkie paper, people are trying to combine the immune agents with chemotherapy drugs, with tyrosine kinase inhibitors, small molecules, and with radiation. And also people are trying to look now at the possible use of immune agents, of multiple immune agents at the same time, trying to combine, for instance, a vaccine with an immune checkpoint drug. Although one of the recent papers in our literature that looked at a combination of vaccine and immune drug did unfortunately not show a benefit.
2 minutes, please.
And this is just, again, going back to the clinicaltrials.gov website, I just put in at the top soft tissue sarcomas in adults and immunotherapy, and it printed out these 3 trials. And it shows you that the website's not perfect because it brought up studies that also involve children. and you can tweak the website to sometimes get more information. But it shows that this is one mechanism you can use to try and look for an immunotherapy trial. And I'll take any questions. Thanks very much.
Great job. Very good. Let's start with some
Thanks so
questions.
much.
Does Keytruda block PDL of all cells, not just cancer?
Well, it may, but that's why you wind up getting these. It works throughout the entire body, so that's why you can get these effects in other places in the body.
Does immunotherapy and targeted treatment work for all leiomyosarcoma patients?
Well, the data on leiomyosarcoma patients, particularly from the SARC-28 trial, was disappointing. There were no responses with leiomyosarcomas. There was also there was another study that was done from Dana-Farber, I think by Dr. George, where they looked specifically at leiomyosarcoma patients, and they did not see responses, at least with the single agent PD-1 drug that they looked at. The Alliance trial that looked at a combination of an immune checkpoint drug and a CTLA drug, ipilimumab, they saw somewhat more responses, but they also saw greater toxicity. So it's been somewhat disappointing so far in leiomyosarcoma, but more work will hopefully improve that.
Are immunotherapy and targeted therapy ever used on patients with no evidence of disease in between scans?
Outside of a clinical trial, I'm not aware of that. Generally, at least in sarcomas, they've been used either people with advanced disease that's progressed, or there have been a few trials looking at it in the neoadjuvant setting, giving it preoperatively, before surgery. But I'm not aware of it being used in the adjuvant setting yet. It's a reasonable thought, and hopefully someone will come up with a clinical trial for it.
How important is absolute lymphocyte count to the success of immunotherapy? At any point below which you would be doubtful of immunotherapy success?
What people have looked at a variety of different markers to try and find out what can predict who will respond to the immune checkpoint drugs. And the SARC-28 trial didn't assess the PD-L1 expression in the tumors, but they went back and looked at it later, and they found that, patients responded irrespective of what the PD-L1 expression was in their tumor. I think it's whether you can mount an immune, whether you can have a response to the drug. People have looked at mutational burden. They've looked at the amount of immune cells in the microenvironment. There's also a recent paper suggesting that the level of B cells, which we don't normally think about for this, that the level of B cells in the area seem to predict who might respond best. So we don't really know at this point, what will cause people to respond to these drugs.
And there was a question, how can you tell the difference between progression and pseudoprogression, or is it possible?
That's challenging, and you basically need to have time. And we talked about RECIST 1.1 for solid tumors. There's a RECIST IR that tweaks the RECIST for immune agents, and you're given more leeway in trying to determine whether someone has true progression when you're giving them one of these immune drugs to sort it out from pseudoprogression.
Okay. And a couple of others. So what is the status of DS3032 I heard about last year? So I don't know if that was for Dr. Algunik or if you know anything. about that. We may have to get back to you on that. And if anybody on the panel knows,
I think we'll get back.
we'll have to get back to you on
Right.
that. Okay, how do you determine necrosis in a large tumor?
The pathologists will do that, and they will, when the tumor is taken out, they will make multiple sections of the tumor and look and assess, how viable the cells are in each region. And this was originally developed for osteosarcoma, and they will give an assessment. They'll say, 90% of the tumor is dead, 95% of the tumor is dead. And in osteosarcoma, those numbers tend to correlate with the patients who are going to do better long-term. When there's poor necrosis, 10 or 20%, those patients are less likely to do well, although they still may.
Okay, let's see. How long should someone stay on immunotherapy?
An excellent question to which we really don't know the answer. Most people now are getting, with sarcomas, are getting it in the setting of advanced disease. If they have a response, the general recommendation has been to treat them as long as they're responding and for up to 2 years. Some patients are being treated beyond the 2-year point. We don't know at this point how long people need to be continued on them.
All right, and then the FDA is fast tracking Annamycin for lung mets in sarcoma. How can I help facilitate opening addition study centers for this drug's clinical trials? I think that's probably for doctors. I think you could contact us directly, and we can see if we can, see if that's something that our center is willing to open. So I think I could speak for the doctors on the panel.